我可能要在这里。对啊，所以你听得到吗？这样听得到吗？这样有吗？没有声音？有。OK。All right. So it's about time. So let's get start today's lecture. And I'm CY from IMI. And the topic of today is defeating antibiotic resistant bacteria, exploring alternative drug targets for a post antibiotic era. All right. So mm, okay. So first of all, I would like to talk outline for today's lecture, and I will divide the lecture into four major parts. The first part, I will talk about how do antibiotics work. And the second part, I would like to introduce you about the antibiotic resistant mechanisms. The third part, we will talk about the AST, antibiotic susceptibility test. And the last part of today's lecture, we focus on the post-antibiotic eras here. So now what? Okay. So let me give you a brief introduction about what was life like before antibiotics. And here, okay. So basically, I would like to let this lecture more interactive. So I would like to ask you a lot of questions. If you know the answer, just feel free to answer a question. Okay. And anyone knows what disease this guy had? What? Nope. It's a bacterial infectious disease, not associated with a virus. Yeah, syphilis. Yeah. So before antibody development, you can see, syphilis, even the syphilis is kind of difficult to treat. Okay. And how about these three? Do you know what disease they had? Just put them, lie on the street. Can you guess what bacterial infectious disease these three men had? Actually, okay, let me just give you the answer. Yeah, because it's associated with mycobacterial tuberculosis infection. But however, we don't have antibiotic for the treatment of this kind of disease. So just let them, lie on the street in case that they will just, like contagious spread to the others. Okay. So this is before antibiotic development. And the third one, even the birth complication, it always is a serious problem, caused by the bacterial infection and also lead to the high mortality rate before antibiotic discovery. Okay. And then the accidental discovery of penicillin in 1928. And anyone know who is he? Yeah, Fleming. Yeah. Okay. How to remove this one? Oh my God. Wait a second. Okay. So it's Alexander Fleming. And you can see here, it's accidental discovery of this antibiotics. So you can see here is a penicillin colony. So there is no bacteria grow close to here. So we call it an inhibition zone. Okay, just like here. You can see the area of inhibition of bacteria growth, just like here. So that's why we will also introduce about the AST, antimicrobial susceptibility test. And you can see an inhibition zone here because the penicillin can release some compound to suppress the growth of bacteria. So that's why here is an inhibition zone. Okay. And then Alexander Fleming just give this penicillin notalent to the chemist wrapper to Wisconsin, University of Wisconsin. So after that, wrapper, he also tried to modify, genetically modify this chain, and then this bacteria can produce a huge amount of penicillin. So that's during the D days, it saved a lot of life for these soldiers. Okay, because the rappers researched at the University of Wisconsin-Madison, they try to modify this chain, and then this chain can produce a huge amount of penicillin. Okay, so I also want to talk about the timelines of the discovery of different antibiotic class. And from here, you can see 1928, the first discovery of penicillin. So it's an important antibiotics. And then the other important drug we call tetracycline. This is very common use in clinical practice. And you can see some drugs such as like cephalosporin and also the trimethoprim and also quinolones. Right now we have 3 or 4 generation of quinolone. We call it a fluoroquinolone. Okay, we will talk about this later. And one of the important antibiotics we call it carbapenem, because a long time ago, carbapenem become a last line of antibiotics for the treatment of MDR, multi-drug resistant bacterial infection. But now carbapenem become the second line of antibiotic for the treatment. Okay, because the rest of antibiotic resistant to carbapenem. Okay, and you can see after 1980, there is only one drug discovered. We call it lipopeptide. Okay. And in addition, I will go on to talk about the mycobacteria or microbial source of antimicrobial agents. And let me give you some introduction about these drugs. And you can see these antibiotics, most of them come from the bacteria or fungi. So for example, you can see gram-positive rats such as Bacillus. And Bacillus subtilis can produce one antibiotic we call bacitracin. And you can also see actinomycetes such as streptomyces. It can also produce important antibiotic we call it chlorphenicol. Okay, and some fungi, it can also produce these antibiotics such as like penicillin or cefalothin. Okay, but 2 of them are not used for the treatment of bacterial infection. It's amphotericin B and also this drug, because these 2 drugs are used for the treatment of fungi infection. Okay, but the rest of them, you can see the other bacteria can produce this drug to kill other bacterial species. Okay, so I want to give you the general concept that most of the antimicrobial agents that come from the microbial source. Okay, and for the antimicrobial therapy, okay, so I want to let you know that we always call it a selective toxicity, just like anti-cancer drug, because sometimes it can, like a selective toxicity can kill or, lead to like a damage of a host tissue. So even the antimicrobial agent, sometimes it can kill harmful microbials, but sometimes it can also lead to the damage of host tissues. Okay. And how about, do you guys know the difference between the antibiotics and also antimicrobial agents? What difference between these 2 terms? Antibiotics and also antimicrobial agents. What difference between these 2 terms? Do you guys have any others? Nope. Okay, antibiotic means natural compounds. Okay, most of them become like is a chemical compounds produced by a microorganism. So we call it natural compounds. They can kill or inhibit the growth of the other microorganism. But however, for the antimicrobial agents, chemicals. So it means that for antimicrobial agents, it can modify the structure from the antibiotics. It's like a synthetic structure. Sometimes we just call it antimicrobial agents. But however, when you read the paper, you can see most of them, they don't care about these terms. They just use antibiotics or use antimicrobial agents. They feel there is no difference between these 2 terms. But originally, for the antibiotic, it means chemical compounds is a natural compound produced by the other microorganism. Okay, so all right. So I also want to talk about the spectrum. For example, you can see for the penicillin, is used for the treatment of gram-positive bacteria, and for the tetracycline, it can be used for the treatment of gram-negative bacteria or gram-positive, or even chlamydia or rickettsia infection. So for this kind of drug, we call it a broad spectrum, okay, or wide spectrum antibiotics. But for penicillin, is a narrow spectrum antibiotics, okay. So you have to know no antibiotic is effective against all microbes, okay. So for example, as I mentioned, for like isoniazid is a narrow spectrum drug. It can be used for treatment of mycobacterial infection. But however, for tetracycline is extended spectrum or wide spectrum antibiotics. It can use for treatment of different species, many different species or bacterial species infection. Okay. And then, oh, wait a second. This is so annoying. I cannot remove this one. All right. Okay, so have any idea how this antibiotic can kill the bacteria? The mechanism through which this antibiotic can kill the bacteria. Do you have any idea? Yeah, the target of the antibiotic, you know, what targets? Cell wall. Okay, and synthesis of proteins. Okay, yeah, and pores, it means like an anti-bacterial membrane. It can disrupt the structure, right? And anything else? Yeah, DNA replication. And say it again. Ribosomal, it means like, but it's very close to the protein synthesis, right? And do you know the central dogma? Yeah, it's from the DNA replication and then transcription. You know, it can synthesize the RNA, and then it's a protein synthesis, right? So it's a translation. So basically, you don't have to remember all of the drug, but I just want to give you a general concept is that there are 6 major targets of antibiotics. The first one, you just have to find the structure first. The first one, the outer, this structure is a cell wall, right? So it can inhibit the synthesis of a cell wall. And then here is a cell membrane. So the second target is a cell membrane, okay? And then you have to remember central dogma. So it can suppress DNA replication, it can suppress RNA transcription, and then it can suppress protein synthesis. Okay, and the last target is folate synthesis. But however, for the folate synthesis, it also associated with nucleic acid synthesis because it can produce target, for example, like tetrahydrofolate. This is to be used for as like a source or for the DNA synthesis. So there are 6 major targets, and the first one is cell wall, and you can see some drug like a beta-lactam drug, like a penicillin, cephalosporin, or carbapenem, and also the vancomycin. Okay, so we will go through all of these drugs later. Okay, so let me talk about the first class of antibiotic is penicillin. How can I remove this one? Okay, so the first target of the antibiotic is cell wall. So the most important antibiotic we call it a beta-lactam drug. So you can see here, there is a beta-lactam ring. Okay, here is a beta-lactam ring structure, just like here. Okay, so there are some major antibiotic we call it penicillin G, and it's require injection, and also penicillin V can be taken orally. Do you know why some antibiotic can be taken orally? Do you know why? Why is antibiotics? Some antibiotics is like you can take orally, but some of them you have to use injection. Do you know why? Basically, is based on the structure. Some of antibiotic is acid tolerant, so it can tolerate to pass stomach. So that's why some of them can be taken orally. Okay, so here these two drugs we call it natural penicillin, or as here mentioned, it's an antibiotic. Okay, it's a natural compound, but here you can see some of them is a semi-synthetic penicillin, such as oxacillin, because oxacillin is resistant to penicillinase. So even this bacteria can produce enzyme, we call it penicillinase. Originally, penicillin can destroy the beta-arrhythmin ring structure here. So it can lead to dysfunction of these antibiotics. But however, we can modify the structure of these antibiotics. So this drug is resistant to penicillinase. Okay. And here we also can modify the structure and lead to these antibiotics show more extended spectrum. So such like ampicillin. Okay, so that's why we need to know the structure of this drug, and then we can modify the structure, and then such as we can cause like extended spectrum or resistant to the other enzymes. Because for the bacteria, you can see here, penicillin here is a beta-arrhythmin ring structure, but if this bacteria can produce a penicillinase, it can disrupt the beta-arrhythmin ring and then cause lead to the dysfunction of these antibiotics. Okay, so for the beta-arrhythmin drug, we can divide into a different class. For example, the most important part is penicillin, such as like penicillin G, penicillin V. I have already introduced to you, and also the other one important drug is cephalosporin. But right now we can divide into a 5 different generation, 5th generation of the cephalosporins, and also the meropenem and also carbapenem. But as I mentioned a long time ago, carbapenem such as imipenem, meropenem, or doripenem. You can see the drug, we always call it the penem. If you see the name penem, it means that this drug belongs to the carbapenem. Okay, so you can see carbapenem is more resistant to the other enzymes. So that's why it's widely used for the treatment of multi-drug resistant bacteria. And here, there is also some drugs. Although this drug cannot kill the bacteria directly, but however, it is a beta-lactamase inhibitor. So it means that even this bacteria can produce some enzyme to disrupt the structure of beta-lactam, you can use a combination therapy. I mean, you can combine the penicillin with carrageenanic acid. Even this bacteria can produce a beta-lactamase. But however, if you combine this drug, you can still kill the pathogen. Okay, but do you know the mechanism why this beta-lactamase can inhibit the function of beta-lactamase? Do you know why? Why this drug can inhibit the function or activity of beta-lactamase? Yeah, you can see the structure here or here is very similar to the beta-lactams. Okay, so it can bind to the beta-lactamase and then it will not let it go. So that's why it can suppress the activity of beta-lactamase. Okay, so if it can let off the beta-lactamase, this beta-lactam drug can kill the bacteria. Okay. So, for example, like a carrageenanic acid or betan is they are very common use for the combination therapy. So for like some check name, you can see here is amoxicillin combined with carrageenanic acid or piperacillin combined with terabetan or the other drug. So that's very common use for the combination therapy. Okay. And here, I also want to let you know some semi-synthetic penicillins or combination therapy. So for some drugs, there are penicillinase resistant penicillin, such as carbapenem. They are very broad spectrum, or meropenem. It's only could be used for treatment of gram-negative bacterial infection. And some of them, based on the structure, we can modify it and make it become the extended spectrum penicillin. Or we can just use a penicillin combined with a beta-lactamase inhibitor to kill the pathogens. Okay, so for the beta-lactam mechanism of action, you can see bacteria require an enzyme, we call it a PBP. here is a transpeptidase. PBP could be used for the synthesis of a cell wall. It can use to link a bridge, amino acid bridge between here, D-alanine, okay? So it can use for the synthesis of a cell wall. But however, for the beta-lactams, it can bind to the transpeptidase active sites here. So it can block activity of transpeptidase and then also lead to the death of bacteria. So this is a mechanism of beta-lactam drug to kill the bacteria. Okay. So the first one is that this antibiotic can suppress cell wall synthesis. And let me to remind you again for the drugs such as beta-lactam, like penicillin, cephalosporin, or carbapenem, or even vancomycin, bacitracin. Okay, this drug could be used for the inhibition of cell wall synthesis. And next one, we are going to talk about the cell membrane. And there are two drugs, we call it polymyxin B or polymyxin E. But however, in clinical practice, we don't use this drug very often because it leads to the damage of your kidney. So we don't want to use these drugs, okay? Okay, so here is a structure of polymyxin B and polymyxin E. Here is a common structure of these two different drugs. But the only one difference is here, the side chain is a little bit different. But for the action of polymyxin, you can see here, polymyxin can bind to the LPS lipopolysaccharide. I think Professor Xueli Huang have already introduced you about the structure of bacteria. So you can see this polymyxin can bind to LPS and then disrupt the structure of outer membrane, and then it can penetrate the peripresent space. And then it can also bind to the inner membrane and then disrupt the structure here. So it can also lead to the death of bacteria. Okay, but now some bacteria, they can modify the structure of LPS, and then you can avoid the binding of polymyxin and then lead to the resistance of these drugs. Okay. Okay, so let's second target of antibiotic is a cell membrane. And next one, I also want to talk about the central dogma, as I asked you, because for central dogma, you can see here is a DNA replication and then transcription for the RNA, and then through a translation, it can synthesize a protein, okay? So the third target of antibiotic is DNA replication. So for the, okay, for this one, don't know why. Okay, it's fine. Okay, so here you can see for the drug, we call it quinolone. Okay, so right now there are fourth generation of quinolone drug, but you can see the name of them always have a fracexin or cysteine. For example, for a second generation of quinolone, such as like ciprofloxacin is very common use for clinical treatment for the UTI infection, urinary tract infection. And here is a third generation of fluoroquinolone, such as levofloxacin. And right now in Taiwan, gemcitabine have already been used for clinical practice. Okay, but for here you can see most of them the structure is very, they are very similar. They have very similar structure here. But the reason why we need to know the function of this structure is that you can modify it. So for example, you can see here, the position 1, this one, position 1 can impact the PK and also drug potency. Okay, and for like position 2, 3, 4, it's associated with binding to the target. So if you can modify the structure of position 2, 3, 4, you probably can improve the affinity of this drug to the target. Okay, so here you can also see position 5 is associated with gram-positive bacteria activity, but position 6 is involved in cell wall penetration. So if you want to improve the penetration activity of this drug, you can just modify the structure here. So that's why it's so important for us to know the structure of these drugs. Okay, but you don't have to remember all of them. I will also talk about this part later. Okay, so during DNA replication, you can see here. Okay, during DNA replication, here is the origin of DNA replication. So it can DNA replicates through the 5' to 3' direction, right? But however, it can lead to the stress here. Okay, so that's why for the DNA drivers, or we call it topoisomers, it can release the stress and then lead to the successful replication of the DNA. But however, for this antibiotic, the third target is here is a topoisomerase. So you can see for the quinolone drug, it can bind to the topoisomerase and then inhibit the activity of topoisomerase. And then it can inhibit the DNA replication and also lead to the SOS response or DNA repair system. But however, if quinolone still bind to this target, and you can see that eventually it can lead to the cell death because this DNA cannot be repaired, okay, and also cannot replicate. Okay, so this is the third target of antibiotics. And the fourth target of antibiotics, the fourth target of antibiotics is RNA polymers. As I mentioned, for central dogma is a DNA replication, RNA transcription, also protein synthesis. So next one is for the RNA synthesis. So here is the structure of rifampin. But right now in clinical practice, for the rifampin is commonly used for the treatment of Mycobacterial tuberculosis infection. Okay, so here you can see DNA-dependent RNA polymers is used for the RNA transcription. But however, for the rifampin, this drug can bind to the beta subunit of RNA polymers, so it can block the mRNA elongation. Okay, so that's a mechanism for the these antibiotics. And for the inhibitor of nucleic acid synthesis, we can divide into the two major parts. The first one, just as I told you, is fluoroquinolone. So fluoroquinolone can be divided into a narrow spectrum. It's a first generation of antibiotic quinolone drug. And then it can also be modified, become a wide spectrum, or such as a second generation or third generation of fluoroquinolone drug. But the other one is drugs such as sulfonamide or trimethoprim. It can be used as an anti-metabolite because it can suppress synthesis of some. Let me introduce you later. It can suppress some synthesis of some metabolite required for the synthesis of DNA. Okay, so here is a fifth target of antibiotics. We call it competitive inhibitors, such as sulfonamide. Okay, so let me introduce you about some action of enzyme inhibitors. The first one is a competitive inhibitors. It means, for example, like this red one and yellow one, they can bind to the same site of this target. So here is a competitive inhibitor. If this one, yellow one, it have already bind to the target enzyme, you can see for this red one, it cannot bind to here. And the other one, we call it a non-competitive inhibitor. It means the binding of this inhibitor can change the structure of this enzyme. So therefore, you can see, this red one cannot bind to this target enzymes. But for the sulfonamide, also trimethoprim, here, the structure for the sulfonamide is very similar to the PAVA here. But you can see for a precursor, and this precursor can through different three major enzymes and then convert into a tetrahydrofolate. And tetrahydrofolate could be used for the DNA synthesis, such as required for the thymine or guanine and adenine synthesis. So here you can see for the sulfonamide or trimethoprim, it can inhibit the function of this enzyme because the structure of these drugs is very similar to the PAVA. So for example, you can see is a competitive inhibitor. For the sulfonamide, it can bind to this enzyme, and thus it can inhibit the PAVA binding to this enzyme. Okay, so that's why we call it competitive inhibitors. Okay. Okay, so the last target, the sixth target of antibiotic is the inhibitor for protein synthesis. So most of these drugs are broad spectrum, but however, some of them show the toxicity problem. For example, like chlorphenicol, it can lead to the damage of your bone marrow. And for like aminoglycosides, such as streptomycin, neomycin, gentamicin, it can also lead to the damage of your kidney, just very similar to the drug for such as polymyxin B and polymyxin E. And for the tetracycline, it can also lead to the problem of your gastrointestinal tract. And macrolide such as like erythromycin is only used for a gram-positive bacteria infection. Okay. Here are some examples of drugs for the inhibition of protein synthesis, but you don't have to remember all of them. For example, you can see here, the first one is the most important one is aminoglycoside drug. And you can see here, aminoglycoside drug, here it can inhibit the initiation. It means the first step of a translation, and it can prevent the formation of an initiation complex. And the other drug, we call it tetracycline. Tetracycline can prevent the amino acid tRNA attached to the A site of a ribosome. Okay, so there are some other drugs such as like clofenical. Because clofenical, it can also inhibit the peptide bond formation. So it can suppress the elongation of peptides. Okay, so but however, you don't have to remember all of them. You just have to know some important drugs such as clofenical, aminoglycoside, and tetracycline, and also macrolide. You just have to know this one. But the detailed mechanism, you don't have to remember all of them. Okay, but here also is some method for you to remember all of these drugs, such as like we always call it like a bi-13 CL50. Because like A means aminoglycoside, T is a tetracycline, the target is 30S ribosome. Okay, and the other one is CL50 means a CELL drug. It's a clofenical erythromycin, lysonodic, and also clindamycin. The target is 50S. Okay, so you don't have to remember all of their mechanism, but you only have to know this drug belongs to the inhibitor of protein synthesis. Okay, but this one is very similar to the previous slide, so I just skip this one. Okay, so let me give you a brief summary of antimicrobial agents. Okay, these antimicrobial agents, how to suppress growth of bacteria. So you have to remember the first target is here, inhibition of cell wall synthesis, such as penicillin, cephalosporin, bacitracin, or vancomycin. And the second target is a membrane, plasma membrane, such as polymyxin B or polymyxin or polymyxin E. And the third target is inhibition of DNA replication, such as fluoroquinolones. And then is suppression of transcription, such as rifampin. And then is inhibition of translation, such as clofenical, erythromycin, tetracycline, or streptomycin. And the sixth target is inhibition of synthesis of some metabolites, such as sulfonamide and also trimethoprim. But however, it can also lead to the inhibition of DNA synthesis. Okay, so you have to remember the 6 target. Okay, but you don't have to remember all of the drug. Okay, so next one is that I would like to talk about the antibiotic resistant mechanisms. And I want to show you some very terrifying story. For example, like here, you can see, For here, you can see the sulfonamide, this drug was introduced in clinical practice in 1935, but however, just within 10 minutes later, you can see the first, antibiotic resistance have already been reported. Okay, and the second one is like a penicillin in clinical practice, but however, just within 5 years later, you can see the antibiotic resistance. And the other drug is methicillin. But methicillin just reintroduced in clinical practice in 1960. But however, you can see just in 1 year, you can see the report of methicillin-resistant pathogens. Okay. And the other drug is vancomycin. This is also very common use in clinical practice for treatment of gram-positive bacterial infection. But however, in 1990s, you can also see the resistance of these antibiotics. Okay. And here, I want to introduce you some important drug-resistant bacteria. And the first one is tuberculosis. And you can see here, for like in 2017, you can see almost 10 million new cases report in the world, and also lead to the 1.6 million deaths. And among these, new cases, you can see almost 558,000 belong to the MDR, multi-drug resistant tuberculosis case. Okay. And you can see it's kind of important, it's an important bacterial infectious disease in some country, such as like Philippines, Indonesia, and also, some countries you can see here, like South Africa or Nigeria. Okay. And the other important pathogen, drug-resistant pathogen, is MRSA. In Taiwan, the MRSA resistance rate is higher than 50%. Okay, but in the United States, it's about 10 to 20%. But in Asian countries, it's higher than 50%. It's very crazy. But you can see here, this MRSA is a most frequent nosocomial pathogen and also resistant to several other antibiotics. So, come again. Okay. Anyway, you can see here for some countries such as like Taiwan or Japan or Singapore, the resistance rate is higher than 50%. Okay, but in the United States, it's about like 10%. Okay. And the other important pathogen is VRE, vancomycin resistant enterococcus. You can see before 2014, you can see there is almost no resistance. It's low resistance of vancomycin. But however, you can see after 2015, you can see a dramatic increase of VRE in Australia. So that's why we need some new drugs, okay, for the complete of or eradication of this pathogen infection. Oop. I just jump to the last one. Wait a second. What's wrong? It's still there. Okay. I want to use this figure to ask you some questions because, as we mentioned, there are some important antibiotics resistant bacteria in the world. But why this bug become the superbug? Can you figure out the mechanism or the reason why this bug become the antibiotic resistant pathogen? So just use this figure. I mean, like for example, you can see, although we try to use several antibiotic for the treatment of this bacteria, but we cannot kill them, right? So the reason is that probably you use the wrong antibiotics, right? But however, you can also see, for example, like here, is over, misuse of these antibiotics. Okay. And for example, you can see some drugs here because when every time, even you have like a cold, you go for a hospital and they will give you some, pills, antibiotics, but I don't think everyone will finish all of them. Probably when you feel better, you just toss off them, right? So that's why you are trying to train this bacteria become antibiotic resistant pathogen. Because if you cannot maintain the concentration of antibiotic in your bloodstream, it means that it becomes like a low concentration. It cannot kill the pathogen. You just try to train them. Use a sublethal dose antibiotic, try to train this bacteria become the resistant strain. Okay. So that's why, right now we have many antibiotic resistant pathogens because you use a wrong antibiotic, it's a misuse of antibiotics. And you don't finish off these antibiotics for your treatment, right? When you feel better, you just, stop to take these pills. Okay. All right. So if you are bacteria and the host try to take some antibiotic to kill you, how can you prevent yourself being killed? Can you get some mechanism? Enzyme production, yeah, because we just talked about like a penicillinase, right? If you can produce a penicillinase, you can just inactivate all of the beta-lactam drugs. And anything else? Yeah, efflux pump. Because this antibiotic, it can penetrate your like a bacterial structure, right? And it can accumulate in the cytosol, and then lead to the death of a pathogen. But however, if you have an active efflux pump, you can pump up all of antibiotics and then reduce the concentration of antibiotic in your cytosol, right? And anything else? Yeah, modification. Yeah, you can inactivate these drugs. Yeah. Anything? Sorry? Oh, stop the uptake. Yeah. So it means like oh, this drug, there must be some channel or transporter, right? For the uptake of this antibiotic. So if you can reduce the expression of this transporter, it means that it can also lead to the antibiotic resistance. Yeah. Anything else? Anything? You mean like you can use like a different enzyme, like alternative? Mm, actually, there are two more mechanisms. And okay, the first one is that this bacteria, they can produce some small peptide, and this peptide can bind to your antibiotic target to prevent the binding of antibiotics. Okay, but however, we can talk about this later. So let me show you some antibiotic resistant mechanism. I think Jason have already mentioned most of them. The first one is that the decrease of the uptake. As I mentioned, there must be some specific channel for the uptake of the antibiotics. But even though this antibiotic can penetrate into the cytosol, but however, if this bacteria can produce an efflux pump, it can just pump out of antibiotic to avoid the accumulation of this drug in the cytosol, right? And in addition, as I mentioned, you can use like enzymes such as penicillinase or beta-lactamase to destroy or inactivate the enzymes. Okay, and in addition, the other one is the alternative enzymes. It means that, for example, sulfonamide and trimethoprim, they can suppress or inhibit the metabolite synthesis, right? But however, if you can use an alternative enzyme, you don't have to use original enzyme. You can produce some alternative enzyme, but this enzyme could not be, binding of this antibiotic, and thus it can lead to the antibiotic resistance. And the other one is a target alteration. For example, fluoroquinolone can bind to the topoisomers, right? But topoisomers, sometimes you can identify the point mutation of these topoisomers. It can reduce the affinity of topoisomer to these antibiotics and then lead to the drug resistance. Okay, so we will talk about this later. But the most important issue is the spread of antibiotic resistance. And do you have any idea about how this antimicrobial could spread to the other bacteria species? What mechanism? Yeah, gene transfer. Yep, but through which kind of mechanisms? Conjugation, right? Yeah, and transformation. Yeah, some of our bacteria we call it nature. We call like a nature transformation because you always uptake the DNA fragment from the environment. Okay, so we call it nature transformation. And the second mechanism is conjugation, uses sex pili. And there's one more mechanism for the antibiotic resistant gene transfer. Virus, yes, bacterial phage. Yeah, there are three major mechanisms for the spread of antibiotic resistance. Okay, so the most important one is plasmid mediated antibiotic resistance. So you can see not only chromosome can produce antibiotic resistant gene, but you can also see some of these genes are located on the plasmid. For example, this plasmid can produce beta-lactamase and lead to the resistance to beta-lactam drugs. Okay, but one issue is that conjugation. Okay, so for here, you can see bacteria can, give the most sulfur gift to the others. It can also lead to the antibiotic resistance, okay? And here, I want to use this as an example to talk about quinolone resistance. For example, you can see here, this plasmid encodes some antibiotic resistant gene. One, we call it QNR. This protein is kind of cool because it's a target protection protein. It's a small peptide. It can bind to the topoisomers, and then it can avoid the binding of antibiotics. So that's why we call it a target protection proteins. Okay, so located on this plasmid, you can also see the other protein we call AAC6'1BCR. This one could be used for the modification of antibiotics and then lead to the inactivation of the drugs. For example, here, you can see it can lead to the modification of the CPO processing, and then you can inactivate these antibiotics. But you can also see some protein, such as like a QEPA or OQXAB. These two proteins are active efflux pumps, so you can just pump out all of the antibiotics. Okay, so it can lead to the antibiotic resistance. But however, if there is any mutation of DNA drivers or topoisomers, it can also lead to the decrease of affinity to antibiotics and also lead to the drug resistance. In addition, there are some channels such as OMC. This one is required for the antibiotic uptake. So if this bacteria can have some mutation of OMC or just reduce the production of OMC, it can also lead to the drug resistance. Okay. Okay, so there are many different mechanisms for the drug resistance. As I mentioned, the first one is a loss of purine. Okay, so if you this bacteria show loss of purine, you can see it lead to the carbapenem resistance because this antibiotic cannot get into the bacteria. Okay, so you can also use bypass targets. As I mentioned, you can use an alternative enzyme and then lead to the resistance. And you can also use a mutation in LPS. As I mentioned, like a polymyxin can bind to LPS and then disrupt the structure of cell membrane, right? So if you can mutation in lipopolysaccharide, or it can also lead to the resistance. And here you can see some ribosomal mutation or modification can also lead to the drug resistance, or just produce some enzyme to degrade the antibiotics, or just overexpress some efflux pump to pump up all of the antibiotics, or just use antibiotic modified enzyme to inactivate this drug, or just target mutation. So there are several mechanisms associated with drug resistance. Okay, but however, if you know all of this mechanism, we can use some combination therapy for the treatment of drug resistance. For example, if this bacteria loss its purine, it means that there is no channel for the uptake of antibiotics, right? But if you can combine some drugs, it can also cause or create a channel. It means that this antibiotic can still enter the cells. Although those drugs cannot kill the bacteria directly, but however, it can create the other channel for the antibiotics. Okay. Okay, so the other resistance mechanism we call intrinsic mechanism of resistance. It means that in clinical practice, you cannot use some specific antibiotic for the treatment because it's a nature resistant. This bacteria is nature resistant to these drugs. For example, like tetracycline, you cannot use for the treatment of Pseudomonas aeruginosa infection because it produces some efflux pump, can just pump out all of the tetracycline. Okay. So that's why we call it intrinsic mechanism of resistance. For example, you can see here for the 3 different antibiotics, A, B, C. But however, only A and B, there is a channel for the uptake of antibiotic A and B, but there is no channel for antibiotic C. So it means that this bacteria is naturally resistant to antibiotic C. Okay, and you can see here, this bacteria can produce some efflux pump. All of the strain in this particular species, they can produce an efflux pump, and specific for the antibiotic B. So you cannot use antibiotic B for treatment of this bacterial infection because you can just pump out all of the antibiotic B. So that's why we call it nature resistance. Okay. And some bacteria we also call it allophone bacteria. Any idea about the allophone bacteria? What is the allophone bacteria? It means that this bacteria originally, it can produce or synthesize some peptidoglycan like a cell wall, but under the stress, this bacteria, it can stop to produce any peptidoglycan, but however, it's still alive. When you remove the stress, it can still produce a peptidoglycan again. So it becomes we call it allophone bacteria. It means originally it's naturally it can produce synthesize some peptidoglycan or cell wall. But however, if you treat this bacteria with like a beta-lactam drug, you want to kill them, So it just stopped to synthesize any peptidoglycan. But when you remove the stress, it can go back to the wild type. It can produce a peptidoglycan to avoid the killing of antibiotics. Okay, so it's very specific term for the bacteria. Okay, so let me give you a summary for the antibiotic acquisition first. Okay, so here you can see how the bacteria can through these three mechanisms and lead to the spread of antibiotic resistant genes. The first one is a transformation, or you can call it natural transformation. Okay, and the second one is a transduction. It means that bacteriophage can also help for the delivery of these antibiotic genes to the other bacterial species. And the third one is a conjugation. Okay, bacteria can use a sex pili to transfer their antibiotic resistant plasmid to the other bacteria. But however, I also want to let you know, do you think for the conjugation, it must be happen in the same bacterial species or different bacterial species? I mean, like if E. coli contain an antibiotic resistant gene, can it just transfer to the Klebsiella pneumonia or Pseudomonas aeruginosa? Can it? Within the different bacterial species, can it happen? Yeah, but I mean, for example, like this one is a E. coli, but this one is a Pseudomonas aeruginosa. Do you think it can happen for? But the truth is yes. Yeah, so that's a big problem right now. Even though the Acinetobacter baumannii, it can just through the conjugation and then transfer the antibiotic resistant plasmid to E. coli. Yeah, that can happen. Okay, so and the other problem for the transduction is that right now, even though we want to use a phage therapy for the treatment of some antibiotic resistant bacteria, right? But we also worry about if this phage can uptake the antibiotic resistant gene, it can also lead to the spread of the antibiotic resistant genes, right? So that would be a big problem in the future. Okay, so that's why it also, due to this reason, it also hamper the development of a phage therapy now. Okay, and okay, I also want to remind you again about the resistant mechanisms. So here you can see it can prevention of a cell wall penetration, and it can also use an efflux pump to pump out of drugs. And also you can modify the target to decrease affinity of this target to the drugs, or you can just degrade or modify the antibiotics. Okay, but the targets, I also want to remind you again, for the antibiotic target include the cell wall and also cell membrane and also DNA synthesis, RNA transcription, protein synthesis, and the last one is for the metabolite synthesis. Okay, so just you want to use this figure to give you a brief summary. All right, so right now in the world there are 6 major species lead to the most important problem for clinical practice. The first one, that's why we call it escape pathogens. Escape means like it can escape from the killing of antibiotics. It can also escape from your immune systems. It means that it's high virulent and it's also antibiotic resistance, most of them. So that's why we call it escape pathogen. Escape pathogen include E. coli, Staphylococcus, Klebsiella pneumonia, and also Acinetobacter baumannii and Pseudomonas aeruginosa, and also Enterococcus faecalis old fashioned. So that's why we just call it escape pathogen. Okay, so there are 2 meanings for this term. It's like escape from the antibiotic, also escape from the immune system. It means that they are high virulent bacteria. Okay. Okay, so I think here is a review to mention about the resistant mechanism of escape pathogen. As I mentioned, like antibiotic inactivation, or like site target modification, or like reduce antibiotic accumulation. But I also want to introduce you one important phenotype of this bacteria is persistence. It means like a bouncing formation. So you can see when this bacteria, planktonic bacteria, attach to the cell surface or attached to the surface, and you can see the isopolysaccharide can start to release, and then you can see the growth of a biofilm. So even in this biofilm structure, there are two different species, and it can also lead to the drug-resistant transfer, gene transfer. Okay, like a conjugation or like a natural transformation could be happen. But however, after maturation, you can see the dispersion of this bacteria. It can also spread to the other place. Okay, just like a cycle. But however, within the biofilm, you can see the antibiotic cannot penetrate the biofilm structure. So that's why it also lead to the drug resistance. Okay. All right, so that is second part for this lecture. And the third part, I would like to give you an introduction about the AST, antimicrobial susceptibility test. And for this part, in clinical practice, we use disk diffusion long time ago. Okay, but however, in Taiwan, sometimes we still use disk diffusion in some small hospital. The only one reason is that I will ask you later. Okay, because basically, if you want to test whether this bacteria is susceptible to some specific antibiotics, you just have to adjust the morpholin. I mean, like a derivative of this bacteria, and then just spread, on the agar plate. Okay, and then you can see here, the second step is just press the antibiotic disc on the agar here. And then the next day, you can determine the inhibition zone. Just like introduction, I talk about the Fleming accidentally discovered the penicillin, right? So you can see here is the inhibition zone. And then you can, based on the CLSI guideline, you can determine whether this drug could be used for treatment of this bacteria or not, okay? And just like here, you can see, for example, this drug is susceptible to this antibiotics. So that's why here is a huge inhibition zone. But however, you cannot use this drug for the treatment of this bacteria because there's no inhibition zone here. It's easy, and the most important is that it's really cheap. Okay, do you know, the price for one disc? One disc, do you know the price? The cost. If you just count, use NTD, it's just probably 0.5 NTD, and you can determine the susceptibility. It's very, very cheap, but just depend on different antibiotics. Some like a carbapenem sometimes more expensive, like $10 for 10 NTD for one disc. But for some commonly used, like a penicillin, probably just one NTD, you can buy a disc. So it's very cheap. So if you want to determine the antibiotic susceptibility test for 20 drugs, it just costs like 20 NTD. So that's why it's very common use long time ago. Okay, but there is a problem for this one, is that you always can only know the susceptibility. You cannot know the MIC, the minimum inhibitory concentration. So there's a problem for this assay. Okay, but right now also have some like specific like a phenotype, we call it heteroresistant. Sometimes you cannot see a clear inhibition zone. Sometimes there is like a small bacteria grow close to here. There's a heteroresistant. And in clinical practice, sometimes you can see here is a growth zone very close to the disc. Okay, but however, there is an inhibition zone here, but there is a growth zone just close to the disc. But however, if you take the bacteria, if there is a growth zone here, around a disc, you assume it must be resistant to this drug, right? But if you take it out and then spray on the agar plate again, you can see the inhibition zone again. But however, there is also a growth zone close to the disc. We call it a heteroresistant. That happens sometimes in clinical practice. Okay. All right, so you can see in clinical practice, sometimes you would just use a dispenser. It saves time. You can just put like a 12 or 15 disc on the agar at the same time. You just have to press here. And then the next day, we just have to measure the inhibition zone, okay? And the other method is like an e-test. We call it strip. But for the e-test, you can see, for example, here is antibiotics here, but you can see the number here. So the reason why e-test sometimes is better than this diffusion because you can see the MIC value. It means like, for example, like this drug, you can see here, it's like a 0.064. Okay, so it means that the inhibition, minimum inhibition concentration of this antibiotic is this one. You can see the MIC value. So compared to the previous one, this one is better because you can see over which concentration you can use to kill the bacteria. But from previous one, you only can know, whether this bacteria is susceptible or resistant to this drug. You cannot know the MIC value. Okay, so that's why this method, you can read the MIC value is better. But however, in clinical practice, we don't use this one. The reason is that it's really expensive. For one strip, it's about 100 NTD. So it means if you want to test for 20, it's like 2,000. So compared to the disc diffusion, this one is kind of expensive. So that's only for the lab. In some lab, we will use this one. Okay. And the other one is a broth dilution. Okay, so broth dilution, it means that you can culture your bacteria in different concentration of antibiotics. For example, this is antibiotic A, and you can just use a 2-fold series dilution from the 256, 128, and then to the 0.5. And the next day, you just inoculate the same amount of bacteria into this wells. Okay, sometimes we just use a 96 well plate, it will be easier. Okay, and then the next day you can see some of them, you can see the bacteria growth here, for example here. So that's why we call the MIC value is 64, because this is a minimum inhibitory concentration for the bacteria growth, right? So you can see the bacteria only grow in a concentration of 32, but it cannot grow in a concentration 64. So that's why we call 64 is a MIC value. But how about the MBC? The minimum bactericidal concentration. What difference between the MIC and MBC? What difference? MIC and MBC. Yep. And it means that in clinical practice, MIC and MBC always like a 2-4 or 4-4 difference. The reason is that here is a MIC, right? Because in this concentration, the bacteria cannot grow next day, right? But if you take out some, like a median, and then put into the median only, and you can see the growth again, the growth of a bacteria again. It means that you only inhibit the bacteria growth. It is not kill the bacteria. For the MBC means that even though you take out from this median to the fresh median, you cannot see the growth. It is a MBC. So for this case, MIC is 64. In clinical practice, MBC always like a 2-fold difference, like a 128 or 4-fold difference. It means that if you use a higher concentration, you can really kill all of the bacteria. Okay, so that's the difference between the MIC and MBC. Okay, MBC is a minimum bactericidal concentration. It kills all of the bacteria. So even though you remove the antibiotic or the stress, the bacteria still cannot grow because they have already all been killed. Okay. Okay, so based on the CLSI guideline, for example, here you can see Enterobacterales. You can see if you use a disc diffusion for these antibiotics is a minocycline. If you use this one, if you like a diameter, okay, it's over the 16. Okay, it means that this one is acceptable. But the range, if in 13 to 15 milliliter, it means that this is intermediate resistant. But if inhibition zone is smaller than 12, it means that it's resistant. So this is based on your disc diffusion result. But if you use a broth dilution, you can see the MIC value, right? So if you use a MIC value is lower than 4 microgram per ml, it means that it's acceptable. But if that one is higher than 16, it means that is resistant. Okay, just depend on which method you use to determine the antibiotic susceptibility. Okay, but right now in clinical practice, we also have some genotypic assays. But what difference? Which one is better? If you use a genotypic assay or phenotypic assay, right now we are talking about a phenotypic assay, right? To see whether there is an inhibition zone, to see the MIC value is a phenotypic assay. But how about some molecular method like a genotypic assay? Which one is better? Which one is better? Yeah, so actually, I would still suggest that use a combination of these two methods because for the genotypic assay, like a molecular assays, you can get a result quickly, right? Yeah, it's faster compared to the phenotype because phenotype always take like 18 to 24 hours. But honestly, in clinical practice, you cannot wait. You will just give an antibiotics. So if you use a molecular method, you can have like a antibiotic. You can assume this bacteria are probably resistant to some drugs because it contains some antibiotic resistant genes, right? You can identify this one just in 2 hours or just in 1 hours. You can give them the antibiotics, accurately. But however, there is a problem. It means that even though this bacteria contains some antibiotic resistant gene, it did not really show antibiotic resistant phenotypes, right? Because even they contain gene, how about the expression level of these genes, right? So that's why I would say that it would be better to combine these two assays, okay, in the future. All right, so the last part we are going to talk about the post-antibiotic era is here. So now I did not give you guys a slide for this part because all of these are unpublished data in my lab, so I did not put into your slides. Okay. For this part, I want to talk about the superbugs first. And I want to give you some number you can see here. For like a cancer, people die because of cancer in 2050 is about 8.2 million, in the world every year. But however, for the antibiotic resistant infections, people die about like 10 million in 2050. So that's why it's an urgent need for, identify new drugs for the treatment of this drug-resistant bacteria infection. Okay. And the other problem is that right now, we just when you go for a doctor, you can see even though you have RTIS, is respiratory tract infection, most of them are caused by a virus. But however, they will just give you an antibiotic first. For like a Streptococcus pyogenes, group A Streptococcus, is a cause of acute pharyngitis only for 15 to 30% of the case. But however, you can see the antibiotics are prescribed in up to 75% of the case. There's an overuse of these antibiotics. So that's a big problem. So that's why in 2050, the WHO have already suggested there will be more than 10 million, people die from the infection caused by the antibiotic-resistant bacteria. So long time ago, we have some weapons or antibiotic for the treatment of some bacterial infection. But however, right now we have some MDR or SDR or PDR. Do you know what difference between these terms? MDR is a multi-drug resistant, right? And how about the SDR and also the PDR? SDR means extensively drug resistance. So it always only susceptible to one or two antibiotics. For a PDR, pan-drug resistant, it means that it's resistant to all antibiotics. For the MDR, it means that it only resistant to 2 or 3 classes of antibiotics. So there's a difference between this definition. MDR only resistant to 2 or 3 classes of antibiotics. SDR means only susceptible to 1 or 2 classes of antibiotic. PDR means resistant to all drugs. Okay, so there's a big problem that right now you can see the a rise of SDR or PDR pathogen in the world. So there will be a big problem. Okay, so let's go back to this slide again. As I mentioned, we have many antibiotics. But the problem is that after 1987, there is almost 30 years is antibiotic discovery voice. You cannot see any new drugs development within, these 30 years. Oh, I should ask you some question. If you are really rich, why don't you want to invest to the antibiotic development? Do you want to invest in drug development? If you are really rich and you have money, do you want to invest in antibiotic development? You want? Why? But most of pharmacy company, they don't want to invest in antibiotic development. Do you know why? Yeah. No, some reason is that the first one is that the cost is high, right? And also take like 10 years or 20 years for the new drug development. The third one, as I mentioned, that for antibiotic resistance, you just development in probably 1 month. If you put this drug in clinical practice for 1 month, you can see an antibiotic resistant drug. So there's why, there's, only a few like a pharmacy company, they still want to invest in this antibiotic development. Most of them, they don't want to do that. Okay, so the first one is a cost, high cost. The second one is a time consuming. And the third one is a resistant development. Okay, so that's a big problem. And here you can see is almost a 33-year gap for the antibiotic, discovery. And there is also like a 55-year gap for gram-negative because fluoroquinolone was developed in 1962, long time ago. So that's a big issue for us right now. So post-antibiotic era is here. So what can we do? So right now, in the United States, previously we have the other plan, is that we ask a senior high school student, they just go outside to, find some new bacteria strain or fungi strain. As I mentioned, most of drugs, they come from the microorganism, right? So they just ask this senior high school student to go to, go outside and then to take some sample back and to test whether their samples contain some bacteria or fungi. They can produce novel antibiotics. Okay, so in addition, some of them just go to identify new bacteria species. And in my lab, we also try to identify some probiotics. It can produce some like bacterial zinc can kill these pathogens. For example, from like a pickle. Okay. All right. So, but I also want to talk about some new drug currently in clinical practice. This is published in this year. Oh, sorry, last year, 2021. So there are still some new drugs, but however, you can see 4 new drugs like cefazidime, avivatin, or ceftaroline and terabatin. But you can see these 4 drugs is a combination therapy. It means like they just combine with beta-lactamase inhibitor. Okay, but there are 4 new drugs. It's a prazomycin. It belongs to aminoglycoside, and also iravicycline, and also temocillin, and also this one. This one is really, really cool. I would like to introduce this one later. Okay, but however, you have to know, as I mentioned, for the carbapenem was considered as a last line for the antibiotic treatment for MDR long time ago. But right now, because the rise of carbapenem-resistant pathogens, so this drug become the second line of antibiotic for treatment of MDR. But one issue is that carbapenem-resistant bacteria, they can produce carbapenemase. Okay, so for example, this is a class A carbapenemase, we call it KPC. It's a Klebsiella pneumonia carbapenemase, identified in Klebsiella pneumonia long time ago. And this is a class D carbapenemase. This is a class B carbapenemase. But however, you can see for like cefazidime, avivatin. Cefazidime is a cephalosporin drug. But if you combine with avivatin, is a beta-lactamase inhibitor. It can be used for the treatment of carbapenem-resistant pathogen. Okay, so for the cefazidime, avivatin introduced in Taiwan about 2 or 3 years ago, but because our insurance system, I think it's really good. Otherwise, we cannot use this drug because for treatment of a patient, in fact, with carbapenem-resistant pathogen, if you use this drug, cefazidime, avivatin, it costs about 10,000 NTD 1 day. It's really, really expensive. And right now, this drug only approved in about 6 to 10 countries in the world because it's high cost. Okay, but I also want to let you know, in Taiwan, in like a TVGH or other hospital or NTU hospital, we just started to use this drug about 1 year ago, but we have already identified many drug-resistant bacteria. Yeah, so that's a big problem. Okay, so I also want to show you some data for this one first. For example, you can see, although this drug just discovered about like 6 years ago, but however, for cefazidime, avivatin have already been reported here. You can see from 2015, is some resistant bacteria have already been reported. Okay, even it's just not, really put into the clinical practice, you can also see the resistant bacteria have already been there. So that is a big problem. Most of the pharmacy company, they don't want to invest the money for antibiotic, discovery, okay? And the other one, this drug is cefidorocol. This one is really, really cool. Okay, let me introduce. Do you guys ever, do you guys know this drug? No? This one is really, really cool. Okay, let me show you. Because when you have an infection, your immune system will create a system we call it a nutrition immunity. Do you know the nutrition immunity? Nutrition immunity means like, for example, for the replication or the growth of bacteria, it needs some like iron or some metals, right? But however, for your immune system, it can secrete some protein to chelate, to bind off these metals, to avoid this metal could be used for replication of pathogens. So we call it a nutrition immunity. Okay, so for the pathogen can release one protein, we call it CD4. And CD4 can bind to this, like metals, and then use some specific transporter to uptake these metals for the growth of a pathogen. It means like a competitor for the host immune system. Okay, but however, this drug, cefidorocol, this is really cool because it can mimic the CD4. It can bind this one and then being transported into the bacteria through its own system because it just mimic the CD4. So that's why I always call it like a story. This story, it just mimic like a CD4, and then it can be uptake by the bacteria transporter. And then it can also lead to the death of a bacteria because it can bind to the PBP and then inhibit the cell wall synthesis. Okay, but this one is, for example, like Acetylbarbeni or Pseudomonas aeruginosa, these two pathogens are more sensitive to this drug because it can uptake it without the binding of the other metals. You can just uptake it. Okay, but however, right now also being some antibiotic resistance have already been reported. The first one is that the mutation of a transporter. If these bacteria have some mutation or decrease of expression of a transporter, you can lead to the resistance to these drugs. In addition, some beta-lactamase, you can find some mutation of a beta-lactamase. You can see here, it can also bind to this drug and also lead to the degrade of this drug. So even though this drug, I think it haven't been approved in Taiwan, but we have already identified some drug-resistant bacteria. Yeah, so this is so crazy. They have already been there. Yeah, okay. So the other one drug haven't been approved, but this one is also quite cool. It's a AAC, antibiotic conjugate drug. Okay, the reason is that for the Staphylococcus aureus, it can hide in the cell. It can hide in the cell. It means like it's an intracellular pathogen. It can hide there. But however, for some antibiotic, it cannot penetrate the host cell. So it means that for this antibiotic, it cannot kill the intracellular pathogens, right? For these antibiotics. So the reason why they want to develop the AAC is that you can see here, the structure of the AAC contains an antibody. This antibody can recognize the structure of Staphylococcus aureus. But however, here is a linker here. And then it can bind hundreds of different antibiotics. Okay, so this antibody can recognize Staphylococcus aureus. And then it can enhance the abscession by the immune cells. But however, when this antibody being uptake into the endosome, you can see there in the endosome there is some like a lysozyme or some enzymes. This enzyme will cleave here. So if this enzyme cleave here, and it can release and activate the antibiotics. So it means that this antibiotic only will be activated when in the cells. So it only kill intracellular pathogen. Okay, so here you can see, for example, this antibody can bind to the Staphylococcus aureus and then being absorption uptake into the host cells. But Staphylococcus aureus can survive in the cell, right? But however, here there is some enzymes. It can degrade the linker and then release the antibiotics. And this antibiotic, can be like a condensed in the endosome and then kill the pathogen. So you only kill the intracellular pathogen. Okay. All right. So there are some new drugs, although some of them haven't been approved in clinical practice, but I think the idea is kind of cool. Okay. So I also want to talk about the other part. The last part is about the research in my lab and also some drug development. Okay. So the research in my lab, we divide into 4 major parts. The first one is bacterial pathogenesis. And we also focus on the gene regulation and also the appearance in pathogenesis. The model organism we use include Helicobacter pylori, also Listeria monocytogenes. And here you can see we want to identify some like new or novel appearance factor involved in the bacterial pathogenesis. We want to see, to determine whether it could be like a candidate for the alternative treatment in the future. So for example, we identify some protein. These protein are small RNA binding protein. But if you can block the activity of this small RNA binding protein, it can reduce the appearance of this pathogen. So right now in my lab, for example, you can see this is a wild type strain, Helicobacter pylori. But if you delete this gene, you can see the motility zone is decreased, and this bacteria also lost flagella. It means that it's low appearance compared to the wild type strain. So in my lab right now, we use multi-omic to determine the function of this novel appearance factors, such as we use transcriptomic, proteomic, or use metabolomic to understand the function of these genes. And the second part of research in my lab is antibiotic resistant. So we cooperate with several hospitals to determine epidemiology survey, and also we want to know the resistant mechanism. So we use some like a pulse field to identify some, to identify whether these resistant bacteria, whether they belong to the same clone, and we also want to determine the resistant mechanisms. So here is an example. You can see we collect the strain from 1997 to 2015. It's almost 19 years. And we want to see the carbapenem non-susceptible acetylbeta strain. You can see the dramatic increase of drug-resistant bacteria in the hospital. All of these strains isolated from patient with bacteremia infection. It means like an invasive infection. Okay. And the third part in my lab, we focus on the bacterial community. So we want to determine the microbiota, association of microbiota with the disease. Because as you know, FMT, the fecal microbiota transplantation, have already been approved for the treatment of Clostridia infection. So that's why we want to know whether some the microbiota composition is associated with the disease development. If we can use probiotics to change the composition of microbiota, probably we can use for serve as like a alternative treatment or prevention of some disease. So we also identify some patients with have a recurrent infections. So let me show you some example. Like here we use microbiota to identify some pathogen is associated with a chronic kidney disease, the CKD. And here you can see some patients. For example, like a patient 1, we isolate 3 strains from the patient 1, but at different time point. It means for example, January, we can isolate this strain. And then March, we isolate the other strain. And then in December, we isolate the other strain from a single patient. But however, you can see the pattern of the 3 strains, they are identical. It means they belong to the same strain. But it's kind of cool because all of these patients have a recurrent urinary tract infection. They just come for to take the medicine for every 3 months. But however, you can see even we give them the antibiotics, but however, we cannot eradicate the E. coli. But you can see every 3 months we can isolate the same strain. So that's why we use a whole genome sequencing to determine the evolution of pathogen. Why this pathogen become more virulent? Why it always can hide in the host, the niche of a host. So we want to identify it. So in the future we can identify some like a method for like a prevention of a recurrent UTI. So I think there are two niches in the host for this pathogen. The first one is that it just hide into your urinary tract intracellularly. But that's why if you give them this patient some antibiotic, you still cannot kill all for pathogen because it have already high in the cells. And the other one, the second niche is that this bacteria just high into your GI tract, gastrointestinal tract, because it just high layer. So every 3 months, it just cause urinary tract infection. So right now, we also try to use metagenomics to identify the host niche of these pathogens. The reason is that we want to know the niche, and if this pathogen just high in the like gut microbiota, if we can give them some probiotics to reduce the number of this pathogen, probably we can prevent the recurrent UTI. Okay. And the fourth part, research part in my lab is translational medicine. So we also focus on the diagnosis and also alternative therapies. So the first one, I will talk about this one later, is drug development associated with MRSA treatment. And the second part is that we cooperate with some company to identify some probiotics, because we identify these probiotics can suppress pathogens. Okay. Okay, so there's four major parts in my lab, and the reason why we want to from like study from the basic research to clinical practice is that if you can find some novel genes involved in the bacterial pathogenesis, you can identify them, and probably they can serve as a candidate for the alternative treatment in the future. So because this is associated with bacterial pathogenesis, so you can see for the motility, if motility is important for the first step for the bacteria attached to the host cell. So if you can block the motility or the adhesion of a pathogen, probably it will be the other alternative treatment. Or you can avoid the synthesis of or production of biofilm, and you can also lead to the decrease of antibiotic resistance. So that's why in my lab we want to focus on from like a basic research to clinical practice. Okay, do you guys need a break, or we just continue? You need a break? Okay, probably we just have 5 minutes. Okay, okay. Okay, so let's get started. I think it just take like 30 minutes, okay? And so in my lab, we are interested in like alternative therapies. For example, this paper we published last year, we want to focus on the like exploring the alternative therapy for a post-antibiotic era. So for example, here there are some new drugs. It cannot kill bacteria, directly. For example, like SPR 741. This one, it cannot kill a pathogen directly, but however, it can disrupt the membrane structure, and then it can, increase the uptake of antibiotics. And in my lab right now, we focus on this one. It's an antibacterial kinase. It's a pesta kinase. I will talk about this one later. Because this kinase have already been shown involved in the beta-lactam resistance, biofilm formation, or appearance. So we want to know whether if we inhibit the activity of this kinase can be like an alternative therapy for the treatment of MRSA. And in addition, some drugs focus on the anti-regulator because if you can, suppress the function of regulator, it can reduce expression of appearance factors. And clone sensing is like a communication, between the different bacteria. So that's why if you can suppress the clone sensing, sometimes it's alternative therapies. And as I mentioned, for the bacterial pathogenesis, bacteria can use a flagella or adhesion to attach to a host cell. So there are some drugs, they want to target the motility or bacterial adhesion. And in addition, some bacteria cause disease because of they can release some toxins. So if you can produce some antibodies, this antibody can block the function or activity of this toxin. It could be the other alternative therapies. Okay. Okay. So in my lab, when I was still in UW medicine, and we focus on this pathogen is a MRSA. Methicillin-resistant Staphylococcus aureus, and I think you guys don't have this slide. And because for MRSA is a commensal and also pathogenic bacteria, it can cause a nosocomial or community-acquired infection. And the most important part is that for the MRSA, it's resistant to almost all beta-lactam drugs. And we identify one pesta kinase, we call it STK1. And we want to know whether we can explore STK1 to augment the beta-lactam activity in MRSA. Okay, just as I mentioned, the MRSA, is higher than 50% in Taiwan, especially in Asian country. So that's why we want to know whether this could be a good target for the treatment of MRSA infection. Okay, and let me give you a brief introduction about pesta kinase. Well, pesta kinase is a penicillin binding protein and serine associated kinase. So you can see here is a pesta domain. Okay, pesta domain could be used to sense a change of environment, and here is a kinase domain. It can, use for the signal transduction. So pesta kinase contain, as I mentioned, like a transmembrane eukaryotic-like serine kinase, and it contains an intracellular kinase domain, also extracellular pesta domains. And pesta kinase have already been shown in Staphylococcus aureus and also Enterococcus mycobacterial tuberculosis. And for mycobacterial tuberculosis is essential. It means that if you can block the activity of pesta kinase, you can just kill the mycobacterial tuberculosis. But however, for Staphylococcus aureus, it's not essential proteins. So even though you block this kinase activity, however, this bacteria will not be, killed. And it has already been shown in cell wall maintenance and also central metabolism, balance, biofilm formation. And the most important part is that it also involved in the intrinsic beta-lactam antibiotic resistance. Okay. So in my previous lab, we use 3 libraries. We contain 1,147 compounds. We try to screen some compounds can suppress the activity of pesta kinase. Okay. And the reason why we want to identify this compound is that, for example, like a wild type strand is a MRSA strand. So you can see this is a MIC value. For the MRSA strand, if you want to inhibit the growth of a MRSA strand, the MIC is about 32 microgram per ml oxacillin. Okay, but however, if you treat the wild type with a GW drug, this is a compound we identify from our screening. And if you combine the wild type with this GW drug, the peptide kinase inhibitor, you can see the MIC value is about 1. So it means that this compound can inhibit the function of peptide kinase, and then it can resensitize a MRSA strand to beta-lactam drug. Okay. Originally, you cannot use oxacillin to kill the MRSA, right? But however, if you treat the bacteria with GW compound, you can use oxacillin to kill this pathogen. It means I resensitize it to beta-lactam drugs. So we also create a STK1 mutant. This is a peptide kinase mutant. But for the peptide kinase mutant, you can see it also shows low MIC value. It's very similar to a wild type tree with a GW drug because this is a peptide kinase inhibitor. So it means that this one, the peptide kinase deletion mutant, is very similar to wild type tree with GW drugs. Okay. So here you can see for the breakpoint based on the CLSI guideline, if your MIC value is lower than 2 microgram per ml, it means it's susceptible. It means that you can use this drug to kill the pathogens. But however, originally the MRSA strand, for example, like MIC to oxacillin or nefacillin is about 16 or 32. But however, if you treat with a GW drug, like a combination therapy, you can see the MIC become 1 and also 2. Okay, you can resensitize them. Okay. And the aim of our study is that we want to know, the first thing is that identify the peptide kinase substrate that is required for beta-lactam resistance. As I mentioned, this is a kinase. So we want to know the downstream signal associated with drug resistance. Okay. And the second thing is that we want to know whether we treat the bacteria with GW drug or peptide kinase inhibitor, we will induce the resistance. As I mentioned, it's really important to know the resistance mechanisms of these drugs. And the third one is that we use an animal model. We want to evaluate the efficacy of STK1 inhibitor combined with beta-lactam drug for the treatment of MRSA infection. Okay. So how does STK1 maintain the beta-lactam resistance? That is a question. So we use a two-pronged approach to identify the pathway downstream of STK1. The first one is arm 1, we use a genetic screening by transposon mutant library. And the arm 2, we use phospholytic proteins. I will talk about this later. So we want to see who overlaps. The first one is that arm 1, the transposon mutant library. We use a Nebraska transposon mutant library. Okay, in this mutant library, we can knock out AV genes in Staphylococcus. So we want to see, for example, if this is a mutant library, we have knocked out 1,902 non-essential genes. And we want to see if we treat with beta-lactam sublethal dose, who will become sensitive. I mean, like originally, it's a MRSA is resistant to nefacillin or oxacillin, right? But if we just knock out one genes, and we want to see who become susceptible to this drug, it means this gene is required for the resistance, right? Okay. And the second arm is that we want to identify the phospholytic proteins. It means we treat the MRSA strand with a sublethal dose of beta-lactam. For example, if we treat with a nefacillin sublethal dose, and we want to see which protein is phospholytic, it means that under the stress of beta-lactam, it will be phospholytic. We assume peptide kinase is involved in the beta-lactam resistance. It can phosphorylate downstream target, right? So we want to know which target will be phosphorylated. Okay, so it could be a target of a pesta kinase. So based on the ARM1, we identify 42 targets. It means these 42 genes are required for the resistance of Staphylococcus aureus to beta-lactams. If you delete any one of them, it becomes susceptible. And the ARM2, we identify 84 targets that will be phosphorylated when you treat the MRSA with sublethal dose of beta-lactam. Okay, so it means that combined together, it means that this protein will be phosphorylated if you treat with beta-lactams, and it's essential for the beta-lactam resistance. Okay, so we identify 5 targets. So these 5 targets are PER-A and PER-B, and PER-A, PER-B are involved in purine synthesis, and SUB and GAB are involved in amino acid catabolism. And the MERG is involved in peptidoglycan synthesis. So that makes sense. This gene is involved in the peptidoglycan synthesis, and this is a target of beta-lactam, right? Okay, so my cooperator, they focus on this part, but I'm interested in this part, the purine synthesis. I want to know whether PER-A and PER-B involved in beta-lactam resistance. Okay? It's kind of complicated, right? Is it okay? Okay, okay. So as I mentioned, we identify 5 candidates. This candidate could be phosphorylated under treatment of beta-lactam, and is essential for the beta-lactam resistance. Okay, so first of all, I want to know. Here, you can see that if you treat the bacteria with oxacillin 8 microgram per ml, but you can see for wild type strain, it's a MRSA strain, it can grow easily because it's resistant. The MIC about 16 or 32, right? So we can see the growth of the wild type easily. But however, for the mutant strain, the blue one, you cannot see the growth of mutant strain in MH broth containing oxacillin 8 microgram per ml. Because as I told you, for the pesta kinase deletion mutant, it have already been resensitized to beta-lactam drug. The MIC from 16 become 1, right? So you can see it can be killed if you just use a sublethal dose, you can kill, okay? But if you give the mutant strain 5 micromolar purine, you can see the growth of a bacteria. So that makes sense. Because in previous one, we identified purine synthesis is involved in beta-lactam resistance. And here, if you we give the bacteria some purine, more purine, it can rescue the phenotypes. Okay, so you can see the growth. If you give them some purine, you can see the growth of bacteria, the mutant strain. It means that it become resistant again. And it means that purine is essential for resistance. Okay? And then we want to know whether we treat a bacteria with GW drug, a pesta kinase inhibitor, it will induce the resistance in the future. Okay, so we treat the bacteria, the pesta kinase mutant strain, we treat them with a beta-lactam drug. We treat the wild type with beta-lactam in combined with pesta kinase inhibitor. Okay, there are 2 different meanings. For the first part, it means that if we can identify any, like a clone can grow, it means that it's resistant to beta-lactam. But for the second one, if we identify some bacteria can grow, it means that it probably is resistant to STK1, the pesta kinase, or is resistant to beta-lactams. That's why we use 2 different strains for this test. Okay, and then we can see the growth of bacteria, and we pick up this colony, and we determine the phenotype, and we also perform whole genome sequencing to identify the mutation of these suppressors. We call it suppressors. It means that suppressor, they contain the second mutation, and this second mutation can alleviate the phenotype change caused by the first mutation. So here, the first mutation is a pesta kinase mutation, right? Pesta kinase mutation, it can lead to the resensitize of this MRSA strain to beta-lactam. But if we have a second mutation, it can rescue its resistant phenotype. It becomes resistant again. So that's why it can grow. Okay, so that's why we call it suppressor mutation. Okay, so based on this one, let me show you some example for the suppressor 1 and suppressor 2. You can see here, the wild type strain, if we treat with 8 microgram per ml oxacillin, it can grow because it's MRSA is resistant, right? But for the mutant strain, it cannot grow. So it makes sense, consistent with our previous study. For the suppressor, it can grow. It means that it has some mutation, and then it leads to the resistance to this drug again. Okay. For RPMI, RPMI is a medium ligand of purine. So for the wild type strain, it can grow because it can synthesize purine by itself, so it can grow. But for the mutant strain, it cannot grow in the RPMI because RPMI contain no purine, so it cannot grow. But however, for suppressors, you can see again, the suppressor 1 and suppressor 2, it can grow in RPMI. So it means that it can rescue the purine synthesis, and it can also rescue the beta-lactam resistance because of a second mutation. Okay, it's kind of complicated. Okay, so we collect more than 100 suppressors, but we just test 20 of them. So for example, like here is a wild type strain, and this is a mutant strain. And here is a STK1 mutant we treat with oxacillin. This is a STK1 mutant we treat nefacillin. And this is a wild type strain we treat with GW drug in combined with oxacillin. And GW drug in combined with nefacillin, we treat with 4 different treatment. But you can see for the wild type is resistant to oxacillin, nefacillin. So this is a MIC value. But however, for the mutant strain, you can see the reduce of MIC value here. But for the mutant strain suppressor, you can see it's high resistant to all these drugs. So it means that the second mutation, it can lead to resistance to GW or pesta kinase or the beta-lactams. So we use a whole genome sequencing to identify the SNPs in these 20 suppressors. And luckily, you can see every group, you can see it identify the same genes, GDPP, and this protein just being reported this year is associated with beta-lactam resistance. So it demonstrates the SNP we identify is correct. They are associated with beta-lactam resistance. But however, the function of the others involved in the beta-lactam resistance or GW resistance is still unclear. So that's why we want to go further to determine the function of this gene in beta-lactam resistant or pesta kinase inhibitor resistant. Okay, so here, the one problem is just as I mentioned, there is always a selective toxicity. So you want to know whether GW it also shows cytotoxicity to cells. So we get these cells, primary cell from a mouse. So we just from their bone, we get these primary cells. And you can see the green color or red color means death of these cells. But for this is a control, you can see the cell grow very well, but some of them also contain show the apoptosis, but it makes sense. Okay, but if you treat with 12 nanomole GW, it show high cytotoxicity, it kill almost all of cells. So it means that it cannot be used in clinical practice. So we cooperate with some of our friends to modify the structure. So that's why I told you, if we can know the structure, we can modify the structure and to show less cytotoxicity, but still contain, the function to resensitize MRSA strain. So for example, here you can see if we modify the structure, even in the concentration 6.25 micromolar, it still can, survive. This cell is still alive. So it means like a reduce of cytotoxicity if you just modify the structure. Okay, so let me show you some video. I cannot use this one. Give me a second. We monitor the cell death. This is a control. Okay, so red and green color means death of cell. So this is a positive control. You can see every hours, you can see the increase of the death of these cells. Also the original GW drugs. But however, if we modify the structure, you can see there's less cytotoxicity compared to the GW. There's no green color or not, much like red color here. So it means that we check the cytotoxicity after for 48 hours. And we check the cytotoxicity, we take a photo every 1 hour. So you can see it's less cytotoxicity compared to the GW drugs. Okay. And then let me show. Okay. But the most important part is that in vivo assay, so we use mouse Staphylococcus aureus bacteremia model to demonstrate the role of pesta kinase. So we infect the mice, and then infect mice with Staphylococcus aureus. The red color is a wild type. The green color is mutant strain. But you can compare the wild type to mutant strain. There's no difference between the virulence. It means that bacterial burden is almost the same from the spleen, liver, and kidney. You can see the bacterial number, bacterial load is almost the same. So it means that pesta kinase is not directly associated with virulence of MRSA strain. But however, for this group, you can see the black one compared to the red one. If we treat the MRSA strain wild type with oxacillin, you can see the bacterial burden is almost the same because it's MRSA strain. So even you treat the bacteria or the mice with oxacillin, it cannot kill these pathogens. Okay, so the burden, the bacterial load is almost the same in the kidney, the black compared to the red one. But however, this is a mutant strain. If you treat the mutant strain with a sublethal dose of oxacillin, you can see the dramatic decrease of a bacterial load. It means that pesta kinase, if in this mutant strain, it becomes a sensitive strain to oxacillin. So that's why you treat with a sublethal dose of oxacillin, you still can resensitize it, and it can kill the mutant strain in vivo. Okay. So, however, we still need to identify some compounds show less toxicity compared to the GW, and we will use an animal model to demonstrate it could be alternative treatment in future. Okay. So right now we have already identified some STK1 subjects such as like PER-M, PER-V, involved in purine synthesis and also involved in the beta-lactam resistance. And we also identify some genes involved in the resistance, probably resistant to GW inhibitor or just resistant to beta-lactams. We have to go further to study it. And someone, our in vivo animal model have already demonstrated if you can inhibit the pesta kinase activity combined with beta-lactam, it still could be used for treatment of Staphylococcus infection. Okay, so the last slide that I want to give you some take-home messaging. The first thing is that the future, if you don't, act now, is that in 2050, it will have like 10 million deaths every year just caused by the infection of multi-drug resistant bacteria. So that's why it's an urgent need to have some new drugs or alternative therapies. Okay, so I think that's all. Yeah. I think that's all for today's lecture. And do you guys have any questions? Any questions?
Hello. Yes, Professor. I have one question. Sometimes doctors prescribe antibiotics for one person for 3 days cycle and for other person 7 days cycle. So I really want to know Is it different among different individuals? Because some people just eat for same problem. Some people are eating for 3 days and they are cured from any infection, but some other people are eating for 7 days. Okay. In short, my question is antibiotics have different cycles for 3 days, 7 days. So,
Yeah, but right now, for example, like a Helicobacter pylori therapies in different country or different hospital, they just try to modify the prescription. So, for example, a long time ago, we always use like a 7 dose, and we use a triple therapy. We use 2 antibiotics combined with bismuth, or like a proton pump inhibitor (PPI) for the treatment of Helicobacter pylori. But just depend on the different hospitals. Sometimes right now for treatment, we just use like 3 days for treatment of Helicobacter pylori, infection. So it's based on the different hospitals. Yeah. But if you can see, there's no improvement or eradication of this pathogen, they will just change the antibiotic use or, change the other drugs. I think that's very common rules, yeah, in clinical practice.
I have one more question. So, doctors are prescribing bacteriostatic drugs for infection, or they are only prescribing bactericidal drugs? I mean, bacteriostatic and bactericidal. So, in clinical practice, doctors are prescribing bacteriostatic drugs because their function is to stop. So, maybe in near future or maybe in late future, bacteria can grow again because their growth was not killed. They were not killed. Their growth was just stopped.
Yeah, honestly, in clinical practice, it's like based on the experience. I mean, like for Israel, we don't have any like MIC data, antibiotic susceptibility test. Even we don't have that one. We just based on the experience. For example, UTI, we just given several sporin, something like that. So there's a possibility that this bacteria can grow again. So that's why sometimes, you can see like for recurrent infection, we have some patients have like a recurrent infection. But however, sometimes just isolate the bacteria from the urine. But 3 months later, you can isolate bacteria from the sputum. Sometimes you can isolate bacteria from the bloodstream. Well, we don't know why, but just because you kill most of the bacteria, sometimes you cannot kill all of them. They just hide into the cells. That's why we always call it like an intracellular community. It means that bacteria can hide in the cell and then form a biofilm intracellularly, so you cannot kill them.
Thank you,
But it
Professor.
depends on the strain.
Okay.
Yeah, if you are lucky, you infect not the virulent strain is fine, but some strains more virulent, it can hide.
Okay, thank you, Professor. I have last question. Can we take probiotics by ourselves or we need to consult doctors first?
You mean
Probiotics.
probiotics?
Yeah. I mean, these are OTC, like over-the-counter drug. Can we buy by ourselves or do we need to go to doctors to prescribe us something?
Yeah, you don't have to ask them for like a prescription. You can just, use that one. But I have to tell you, the truth is that even the same probiotics could not be like a benefit for everyone. It's not benefit for everyone.
It varies among individuals.
Because we always mention like, the effect is based on the host factor, bacterial factor, and also environment factor, right? So even though you can take the same probiotics, but however, you cannot have the same effect for everyone. Yeah.
Thank you, Professor.
I have a question. When you are like doing the research with the mutated, like the gene that you already know that can be involved and you are testing it, can it be possible that one gene is necessary for other gene to express, and that's why you cannot understand if this gene or both genes are dependent for expressing some protein that can be pathogen or can be involved in the resistance mechanism?
You mean like if you just delete one gene and sometimes it's correlated with a second gene effect?
Yeah, like maybe the expression of this gene depends on the expression of the other one.
Yeah.
Can you target both at the same time, or how can you just
Yeah, we can study both of them at the same time, meaning like a double mutant. Yeah. So the strategy is that for our first arm, we just want to identify which gene is important for the drug resistance, right? So we use Nebraska mutant library. It means like we have already knocked out every genes. Okay. So, but for the suppressor mutation, it means that we want to identify the other genes probably also involved in the resistance. So it's a second mutation. But however, we still have to evaluate the connection between the second mutation to the PASDA kinase. But just as you mentioned, sometimes you just delete one gene, you cannot see the effect. Sometimes you have to delete 2 or 3 more genes to see the effect. Because sometimes it's not only one copy. I mean, like for you delete one gene, but there are some copies, the other gene, they can compensate the function. So even though you just delete one of them, you cannot see the effect. You have to delete probably 2 or 3 of them. Yeah, so there's a limit of our study. We just delete one gene every time. Yeah. But it's kind of complicated to study the gene regulation. Yeah, because right now, still some gene, even in bacteria, we don't know the function of these genes. And some of them are small RNA. Yeah, because in eukaryote, I think all of you know it's like a microRNA, right? But however, in bacteria right now, it's more like a small RNA. The function is unclear. Also, like a ribose switch, even this RNA, but sometimes they don't have function. They could not be translated into a protein, but they still have some regulatory function. So it's really complicated to study the functions, yeah, of these novel genes. Yeah.
Professor, I'm just want to clarify because isn't it that when you do transposon mutagenesis, you do that to randomly mutate genes on a particular strain, and that was used by the Nebraska transposon library. So that gene are already established, right? So meaning to say, when you order something, a strain on the Nebraska transposon, you know the specific gene that was mutated.
Yes. Yeah.
Okay.
That's why they can just sell it. I mean, like for Nebraska mutant library, they have already cloned in, and then they have already determined, for example, the clone number 1, they know which gene have already been inserted and inactivated. So they just sell this mutant library to others because they have, whole mutant.
How do they know the specific? They do whole genome?
No, I think because there are some methods. For example, for transposon mutant library, always like a transposon can jump into like some specific site, right? But there are also some limit for the transposon mutant library. The first one is that there is always hotspot. So I mean, like for like a gene A, you can identify like 5 transposons, like 5 clones contain the A gene have a transposon insert. But for the gene B, probably just one. So that's why we call the gene A is a hotspot. It's kind of, we don't know why, but it's like a hotspot. Transposon always just jump into the A gene to inactivate the A gene. Okay, so there is a limit. And the second limit is that for this transposon mutant library, you can only get the non-essential genes. Because for essential gene, it's no way for you. Because if you knock out or like inactivate this gene, bacteria have already died. Yeah. So you cannot, evaluate the function of essential gene. So there's a limit for the transposon mutant library. But if you have a transposon insert, we just have to do like a cloning or do the PCR, something like that. Like a digestion or like a circular and to sequence, you can see the site, the gene locus, where the transposon have already insert. Yeah.
Thank you, Professor.
Yeah. No? Okay. No? No more questions? Okay. Okay. Yeah. Okay. I think that's all for today's lecture. If you have any questions, feel free to contact me. Okay. Yeah. Yeah.
Online, that is synchronous, right? Not pre-recorded. so we can go or just stay in our lab or something.